Hello everyone, in this video I will briefly talk about my RU program experience. First of all, a little bit about myself. My name is Cruz Delgado, and for this summer program I had to fly all the way from La Isla del Encanto, Puerto Rico. Even though Ponce, as you can see in red, is my hometown, I am a chemical engineering undergraduate student at the University of Puerto Rico in the city of Mayagüez. During this summer, I have been working at the Biomaterials and Drug Delivery Laboratory under the mentorship of Dr. Blanca Sharma and alongside the doctoral student Susan Lightsey in a project focused on the characterization of lung cancer cell growth in hypoxia-inducing cryogels. As you can see in the photos, the Sharma Lab is more than a group of researchers. It's a team, literally. We won second place on the BME's department's field day. We are interested in studying cancer because, according to the World Health Organization, it is the second leading cause of death worldwide. Our work was focused on solid tumors and on one of many cancer hallmarks, hypoxia. Hypoxia is defined as oxygen deficiency, less than 5% of oxygen to be precise, and it is associated with the poor vascularization that makes difficult for the bloodstream to reach the core of the tumor. A better understanding of this solid tumor barrier is needed in order to develop more reliable anti-cancer drug delivery systems. So, in this project, we work with an in vitro 3D scaffold to mimic the microenvironment of tumors, and this way study cell behavior in hypoxic conditions. For this, we use hypoxia-inducing cryogels, which are made out of methacrylated hyaluronic acid and designed by our collaborators at Northeastern University. During the past weeks, we completed all of the objectives for these projects, adjusting the adhesion time until finding an optimal one, characterizing cancer cell growth over time, and determining the effector to target cell ratio for further immune cells infiltration studies. To achieve this, I had to learn new laboratory techniques and protocols, such as double-stranded or dsDNA assays, MTS assays, ELISAs assays, confocal microscope imaging, among others. I also improved interpreting and analyzing data results, but most of all, I gained a better understanding of how scientists' work is applied to real-life problems. But not all the summer was about doing research. I also got to do a lot of amazing things, such as tubing down a river at the springs, going to a World Equestrian Center, taking salsa classes, and visiting the Kennedy Space Center. Even though all of these experiences were great, the best part of the summer were the people that I met, the valuable relationships that I built and that I will cherish all of my life. I want to thank the National Science Foundation for the funding and acknowledge my home school, University of Puerto Rico at Mayagüez, the University of Florida, and the Sharma Lab, in particular to Dr. Sharma and Susan Lightsey, because without their support, this would haven't been possible.